GlycoMira was founded in about 2010 as a licensee from the University of Utah for some technology developed by one of their leading faculty members. And the company's charge was to create a heparin mimic. We've known for, I say we, the community of science and medicine has known for many years that heparin has some very pronounced anti-inflammatory properties, but due to its anticoagulant activity, it's seeing very little clinical use outside of a blood thinner. So the goal was to create a mimic that would have the anti-inflammatory properties, but not the same anticoagulant activities. And to that extent, the company began to develop some molecules based upon sodium hyaluronate, short chain polymer, by substituting on each of the dimers, several of the hydroxides were substituted with sulfates. This created a new unknown molecule that indeed had very pronounced anti-inflammatory properties, but very little anticoagulant activities. As you well know, over the past decade, immune modulation or checkpoint inhibitors have become powerful tools, but they're not without some shortcomings. There's some potential for serious side effects from infusion as well as life-threatening autoimmune reactions. And in addition, they're obscenely expensive. A course of therapy can exceed $150,000, and even with copayment, oftentimes the copayment is $60,000 or more. Our compound is not a biologic. It's a small molecule therapeutic. It's very inexpensive to make. It's very stable. And what it does in a global sense is that it inhibits several pathways in the processes of the tumor microenvironment to enhance solid tumor treatments while also adversely or mitigating the adverse effect arising from radiation. On the tumor side, it blocks CXCL12 binding, it blocks RAGE, it blocks VEGF. So it's truly a multimodal immune modulator, as opposed to most of the current are, single targets. But in addition to this, it also blocks the cytokine storm that comes in and creates the damage to the healthy tissue that occurs as a result of radiation. So our first indication, we looked at this, and our first indication for this is for the treatment of head and neck cancers, obviously from the reducing tumors. But as you may know, a significant number of these patients develop severe mucositis. They come to the clinic 5 days a week for 5 to 7 weeks to receive treatment. And mucositis is a serious side effect, oftentimes leading to cessation in treatment, to oral lesions, to feeding tubes, to narcotics. It's not just a trivial effect of the treatment. So the combination of tumor reduction and mitigation of oral mucositis made head and neck cancer the appropriate first indication for GM-1111. We've completed through the SBIR program, we completed all of the requisite preclinical animal studies. We submitted an IND to CDER, Oncology Division 2, in January. And first time through, they approved us for phase 1A clinical studies. These studies will be done at the Virginia Commonwealth Institute, the Massey Cancer Center in Richmond, Virginia. The reason for that is some of their researchers had done some really nice preclinical work on showing that GM-1111's effects were not limited to head and neck cancers, but also effective in a number of different cancer models, not in actual animal studies, but in organoid models. So with that FDA approval, we're in the process of seeking funds to finance the first phase 1A study at Virginia.